So as Mary and Grace yesterday tell us that inflammation and fibrosis are tightly regulated for the appropriate myocardial repair during heart failure. So excessive fibrosis are mostly associated with cardiac remodeling, and it is a key component for heart failure. And chronic inflammation promotes cardiac fibrosis in many of the cardiovascular diseases. During excessive cardiac stress, the resident fibroblasts they got activated by the inflammatory cells using the pro-inflammatory cytokine or pro-fibrotic factors, which is secreted by most of the inflammatory cells. They activate these resident fibroblasts to become active fibroblasts, what we call myofibroblasts. And these myofibroblasts actually leads the process of cardiac remodeling. And inflammation is the most important factor or chelator to enhance this cardiac remodeling. Therefore, we are thinking that if we eliminate an inflammatory triggers, it will be very important to regulate the activation of cardiac fibroblast, and we can rescue the bad remodeling in the heart. So we are working with the interleukin-10 since last 15 years, and IL-10 is a pleiotropic anti-inflammatory cytokine which drive Th2 kind of inflammatory response. Its major function is as an anti-inflammatory cytokine. In addition to its role on inflammation, it enhances cell differentiation. It also enhances survival. The cardiovascular specific response of IL-10, you can see here, not many papers published, but few papers indicates that IL-10 inhibits post-injury TNF-mediated vascular smooth muscle proliferation. It can inhibit the attachment of monocytes with endothelial cells, and therefore it enhances the endothelial function in myocardium. It also reduces TNF production in chronic heart failure patients. In our previous study, we found that when we treat the wild type and IL-10 knockout mice with isoproterenol, we found that the IL-10 knockout mice have like exaggerated fibrotic response, and at that time, not a single study indicates the role of interleukin-10 with the matrix remodeling. So it was really surprising for us that how IL-10 can regulate the cardiac fibrosis. Most of the literature at that moment was it's related to cardiac myocyte proliferation, cardiac myocyte survival, or macrophage deactivation. So we wanted to pursue this study. So we took wild-type mice and we induced TAC. We wanted to know like this is only with the isoproterenol because or with the other heart failure model. And we found that when we injected interleukin-10 in the mice after TAC, we found that the TAC-induced cardiac fibrosis was significantly inhibited by interleukin-10, and it's improved cardiac function as well. So this was very significant finding for us, and we wanted to pursue the molecular mechanism, how IL-10 regulate this process. So during this study, I was in Dr. Raj Kishore's lab at Chicago, and his lab was totally dedicated for stem cell study. And when I was going for the lab meeting, everyone present, stem cell, and I was studying the basic heart failure biology. So I was thinking that there should be some stem cell niche or some other cell source for cardiac fibrosis as well. And we know that interleukin-10 regulate the mobilization of certain progenitor cells from bone marrow. So we thought that maybe some bone marrow cells can participate in the fibrosis process, and IL-10 might regulate their mobilization. So initially we thought that the major portion of cardiac fibrosis or myofibroblast is mediated by cardiac fibroblast by activation of macrophages. However, some subsets of bone marrow cells might mobilize from bone marrow into the myocardium, and we are thinking that IL-10 might inhibit their mobilization or their activation into the myocardium, and therefore they are regulating this fibrotic response. I started searching the literature and found like a couple of papers which indicates that some progenitor for fibroblast, they are mobilized into the myocardium. They call them fibroblast progenitor cells. These cells express these cells express the stem cell markers like c-KIT, SCA1, CXCR4, and exclusively express Prominin 1 on their surface. They are common progenitor for monocyte macrophage as well as myofibroblast. They nicely present this data that when they treat these cells in culture discs with TGF-beta, they express the fibroblast like marker like fibronectin and alpha SMA, as well as when they treat them with the CSF, they also become myocytes. They suggest that they are common progenitor for fibroblast as well as monocyte macrophage, but they cannot make cardiac myocytes. So initially we start our hypothesis that during initial stress on heart, the cardiac cells, macrophage or cardiac fibroblasts, they send the signal to bone marrow to release these cells because during early phase of stress, heart need to get repair appropriately, and they don't discriminate which cells contribute for their repair, but they want, so these bone marrow cells, they mobilize into the myocardium, and in myocardium, because of presence of inflammatory and profibrotic milieu, they get differentiated and participate or contribute for cardiac fibrosis. And we are thinking that IL-10 either inhibit their mobilization into the myocardium or at least inhibit their activation to participate in this process. So our initial hypothesis was IL-10 inhibits bone marrow fibroblast progenitor cells, mediated cardiac fibrosis in pressure-overloaded myocardium. As a proof of concept experiment, we took wild-type and IL-10 knockout mice. We induced pressure overload in these mice by transaortic constriction surgery. On day 5, we collected blood as well as heart and did FACS to track these cells both in blood as well as in heart. So our data, FACS data, indicates that both in wild-type as well as in IL-10 knockout mice, these cells are mobilized into the myocardium. Interestingly, we found that both in blood as well as in the heart, it indicates that IL-10 knockout mice after TAC have extensively more population of these specific cell population. It suggests that these IL-10 tightly regulate the mobilization of these cells into the myocardium. To further confirm this data, we took the approach of bone marrow transplantation. So in this approach, we took IL-10 knockout mice and we replaced its bone marrow with the GFP with the wild-type mice, which is GFP labeled cells. So all the bone marrow of these mice are GFP labeled. So when we repopulate its bone marrow, so its bone marrow should be GFP labeled. After 5 weeks of repopulation of bone marrow, we induce pressure overload by transaortic surgery, and day 5, we took blood to track those cells, whether these cells are mobilized into the myocardium or not. And we terminate our experiment on day 28 after final echocardiography and collected heart for histology as well as immunocytochemistry analysis to see whether these cells are participated in fibrosis or not. So 5 days FACS data clearly indicate that wild-type mice have more of these cells. IL-10 knockout mice have even higher number of cells in bloodstream. The most interesting data for us is like when we try in the chimeric mice, the IL-10 knockout mice which have like wild-type bone marrow, the mobilization of these specific cell population was significantly reduced. It indicates that IL-10, only repopulation of IL-10 positive bone marrow in the IL-10 knockout mice significantly retain those cells into the bone marrow. They don't let them release from bone marrow. We also took the heart sample on day 5 to see whether these cells are recruited on the heart. So all the green cells, they are come from bone marrow, and few of those yellow cells, which is Prominin 1 and GFP staining, indicates that these are the cells which is Prominin positive cells. They are arrived into the myocardium, although their number is limited because of the wild type marrow in these mice. On day 28, we co-stain, we did the staining for the fibroblast marker together with GFP, And all the GFP cells over here is the bone marrow cells. The red cells are the resident fibroblast. However, the yellow cells indicates that the alpha SMA positive NGFP positive cells clearly indicate that these SMA positive cells, they derive from the bone marrow and participate in the process of fibrosis. Because myofibroblasts have not a dedicated marker, so we further stain these sections with the collagen, and we also found that the GFP positive, some of the GFP positive cells, they co-stain with the collagen. So these data indicates that these cells, they got recruited into the myocardium and they can differentiate to become fibroblast-like cells. We did the mesenteric staining. As I showed you previously, that IL-10 knockout mice after TAC have more fibrosis. However, because of the reduced mobilization of these progenitor cell population in chimeric mice in the heart, we found a significant reduction in cardiac fibrosis in these mice. You can clearly see over there, most of the interstitial fibrosis was gone. We also did the functional analysis of these mice heart, and you can appreciate that the chimeric mice, the heart function was better as compared to the IL-10 knockout mice after TAC. It's indicate that the inhibition or reduced recruitment of these specific bone marrow population into the myocardium significantly improve cardiac function by inhibiting cardiac fibrosis. To tease out the molecular mechanism, we culture those cells in culture dish and try to induce their differentiation to become myofibroblasts. So we treat these cells by TGF-beta, and you can see most of the Prominin 1 positive cells are differentiated and take the myofibroblast-like shape and express alpha SMA. However, the cells which were co-exposed with the IL-10 together with TGF-beta, their differentiation was significantly inhibited. Further, the fibrotic gene expression was significantly inhibited in the mice, in the cells which were co-treated with TGF-beta together with IL-10. So these data clearly indicate that IL-10 not only inhibit their mobilization into the myocardium, but also inhibit their differentiation to become myofibroblast. We also did the protein estimation in these cells. We measured the alpha SMA level after TGF and IL-10 treatment. You can see the TGF-induced expression of alpha SMA was significantly reduced with the IL-10 treated cells, and the TGF downstream signaling target was inhibited as well in these cultured cells after IL-10 treatment. These data clearly indicate the differentiation was significantly reduced. So literature indicates that the differentiation of or activation of cardiac fibroblast is highly dependent on certain microRNAs. So we also did the microRNA array in these cells, wild type and IL-10 knockout FPCs, and found that the level of pro-fibrotic microRNA was significantly higher in the FPCs of IL-10 knockout mice. We did the in vitro analysis after TGF-beta treatment, and we found that the MIR21, MIR145, which is highly activated during fibrotic heart, was significantly upregulated after TGF-beta treatment, and IL-10 inhibit their expression in these cells. To further know the mechanism, it's a very busy slide. I would like to keep your attention on these bars, like after TGF treatment, the MIR21 was significantly upregulated, and IL-10 inhibit that TGF-beta induced upregulation. However, the cells where we treated MIR21 mimic, which further activate the level of MIR in these cells. The last one is like these cells were treated with the MIR21 mimic, IL-10, and TGF-beta. So it indicates that when we overexpress MIR21, the effect of IL-10 was significantly abolished. It clearly suggests us that IL-10 might work via MIR21 inhibition in the system for activation of these cells. And these data were further confirmed for the functional data where we measured the collagen 1 as well as TGF-beta level. The IL-10 mediated inhibition of these fibrotic marker was abolished when we treat them with a mimic. So summary of the first part of my talk. So I tried to convince you that IL-10 regulates fibroblast progenitor cell mobilization from bone marrow into the myocardium. In wild-type bone marrow transplantation experiment, we found that we confirmed that the IL-10 regulate their mobilization and regulate the fibrotic process in stressed myocardium. And the transplantation of bone marrow significantly reduced the cardiac fibrosis and improved the cardiac function. IL-10 inhibit TGF-beta induced transdifferentiation of fibroblast progenitor cell activation in cultured cell. And we propose here that IL-10 might inhibit the TGF-beta SMAT signaling and regulate the mRNA 21 maturation in the system. So this is the cartoon I showed you earlier, that here we fill this part that the activation of these cells were mediated by MIR21 in TAC system, and IL-10 might inhibit this process. So when I submitted this paper to CERC, for my R1 projects, so they questioned me that you are talking about some bone marrow cells, which is like less than 1% in population, and you are showing their physiological response is so immense. So how you can justify that only 1% of bone marrow cells can lead the fibrotic pathology? So during that time, from now to 4 or 5 years ago, it is well debated that the stem cell therapy and controversy with stem cell therapy. So at that moment, we thought that mostly the Marron group, they showed that the stem cell mainly function via their paracrine signaling, and most of the progenitor cells are biologically very active. They secrete a lot of growth factors, which leads their biological functioning. So we are thinking that although our cells are less in number, but they are highly active cells, and they might secrete some growth factors which is related to fibrosis, and they activate the resident fibroblasts to activate the intensity of cardiac fibrosis in IL-10 knockout mice. So our hypothesis over here is that IL-10 knockout FPC might secrete exosomes packed with the profibrotic factor, which activate the resident fibroblast to lead the cardiac fibrosis in IL-10 knockout mice. So I will give you a brief introduction about exosomes. So exosomes are biological entities for communication between cells. They are less than 150 nanometer in size. The beneficial effect of stem cell, mostly for the stem cell and progenitor cells, are mainly mediated by exosomes. And exosomes, the most important point over here is that exosomes mirror the behavior of their cells of origin. If you isolate exosomes from cardiac myocytes, they will have factors linked with the cardiac myocyte biology compared to if you isolate exosomes from endothelial cells or macrophages. Or if you isolate exosomes from diabetic cardiac myocytes, so they will have like phenotype or factors which mimic the phenotype of diabetic cardiac myocytes. So here's some characterization data. So our exosomes were less than 100 nanometer in size, but we measured with the nanosite as well as DLS site scattering. We also did the electron microscopy. We found nice double membrane structure. And we labeled the protein as well as RNA using green dye and red dye, and found that the exosomes are enriched with the protein and RNA. Other people also showed that they are enriched with the long non-coding RNA, lipid, and some other genetic material. So initially, we screened our exosomes to see whether they have the same factor what we found in the fibroblast progenitor cells. So we took the wild type as well as IL-10 knockout FPC exosome, and did the fibrosis-associated microRNA array, and we found that MYR21, MYR27, MYR145 are highly enriched in these exosomes as well. We also did the mass spec of these exosomes, and we found that the protein we found in these exosomes are highly enriched and associated with the protein which leads the fibrotic signaling process. So for proof of concept experiment, I always do a proof of concept experiment before lead for the big study. So I took those exosome, culture them, those cells, culture them for 48 hours and collect the exosomes, and then isolate and put those exosomes on isolated mouse hard cardiac fibroblasts to induce their differentiation. If they have those factors, they should induce their differentiation. So these are the wild-type mouse hard cardiac fibroblasts. They were treated with the wild-type as well as IL-10 knockout mice FPC-derived exosomes. And these are the RNA data, the qPCR data for collagen 1, collagen 3, and alpha SMA. So you can see over here, when we treat those cells with these TGF, as we expected, the level of these profibrotic RNA was increased. When we treat those fibroblasts with the wild-type exosome, the expression of these fibrotic RNA was not significantly increased, although it's increased, but not significantly increased. However, the cells which were treated with the IL-10 knockout mice-derived exosomes, the fibrotic gene expression was tremendously increased, clearly suggests that the factor which is packed in these exosomes, they can lead these fibroblast activation to become more fibroblast-like cells. We further confirmed these data with the immunostaining for alpha SMA, and we found that the TGF its own can activate their differentiation to become myofibroblast. However, wild-type treated fibroblasts, they have less expression of these alpha SMA expression. The cells which were treated with the exosome derived from IL-10 knockout mice FPC exosome, a tremendous amount of alpha SMA expression, and their shape was like activated cardiac fibroblasts. So these are really interesting and encouraging data. We found that IL-10 knockout FPC-derived exosome alone can lead the activation of resident fibroblasts. However, in disease condition, they will be more activated. We also did the microRNA 21 expression, whether these exosomes can transfer those microRNA 21 to these fibroblast cells. So these are the resident cardiac fibroblasts after different treatment, and you can see like this is the IL-10 knockout exosome, and this is the IL-10 knockout together with TGF-beta. You can see the level of these MYR21 is tremendously increased after the exosome treatment. So it's clearly indicate that these cells are enriched with the profibrotic factors, and they can transfer those profibrotic factor to the resident fibroblasts to activate them to enhance the cardiac remodeling during disease condition. So here I will summarize my talk, like bone marrow FPC, the secrete exosomes. IL-10 knockout FPC-derived exosomes are enriched in fibrosis-associated microRNA and proteins. The IL-10 knockout FPC-derived exosome trigger cardiac fibroblast transdifferentiation to become myofibroblast, and this function is highly dependent on MYR21 expression. We are investigating the molecular mechanism. We are planning to take these exosomes from IL-10 knockout mice and transplant these exosomes into the wild-type and knockout mice after TAC and see their physiology. And we will try to repair those exosomes by removing those microRNA. We will try to design our exosomes with better factors and then transplant those exosomes in diseased mice to see the beneficial effect after TAC. So here, the final conclusion slide, like fibroblast, after stress, they can get activated to become myofibroblast for fibrosis. We try to show that the bone marrow cells, they recruited into the myocardium, and they themselves can be a myofibroblast, but their population is very low. However, because they're highly active, so they can secrete certain factors packed in exosomes, and they can increase this visual cycle for the activation of resident fibroblast to enhance the fibrotic response in IL-10 knockout mice. And by putting IL-10 in this scenario, we can inhibit their mobilization as well as differentiation to secrete the better exosome to reduce the bad remodeling in the heart. With that, I would like to thank the people who helped for this one. So I recently moved to UAB in last 6 months. So most of the study we did, it was in Temple University, in Dr. Rajkishore's lab. We are still collaborating. The other collaborator from UAB, like Sumant, Martin, and Prasanna Krishnamurthy for good discussion on these topics. And I'm happy to answer any question. Thank you.